रामचंद्रन प्लॉट को समझते हैं। ये प्रोटीन संरचना दिखाता है। ये स्टेरिक हिंड्रेंस बताता है। ये बहुत महत्वपूर्ण है। To understand the Ramachandran plot, we must examine the peptide backbone. Resonance makes the peptide bond rigid and planar, leaving rotation to phi and psi.
The universe feels boundless, but if you look closely enough, the biological machinery keeping you alive operates under restrictive geometric laws. When a cell builds a protein, it strings together a polypeptide chain. In pure mathematics, a chain like this could theoretically bend into countless configurations. But biology is constrained by physics. Atoms are solid objects, and they take up space. Every atom has a fixed physical boundary called its van der Waals radius. When two non-bonded atoms are pushed too close together, their electron clouds repel each other. To avoid structural collapse, a folding protein cannot bend at random. It must navigate these physical boundaries with absolute precision. In 1963, an Indian biophysicist named G.N. Ramachandran set out to map these exact physical boundaries. He and his team were trying to build a stable mathematical model for collagen, a complex triple helix protein. But every time they adjusted the structure, they ran into a severe roadblock. Their equations showed atoms occupying the exact same physical space. In physical chemistry, this is called steric clash or steric hindrance. It is a physical collision that makes a specific molecular shape impossible. To find a way around this, Ramachandran applied a hard sphere approximation. By treating every atom as a rigid sphere with a known radius, his team calculated the exact rotational angles that caused collisions and the angles that were safe. Ramachandran relied on pure geometry to define these limits. He successfully plotted the absolute physical boundaries of molecular movement. In the early 1950s, physical chemists Linus Pauling and Robert Corey demonstrated that the backbone of a protein is bound by strict geometric constraints. This diagram shows why. The carbon-nitrogen linkage has a delocalized double bond character, locking 6 core atoms into a rigid, flat plane, making rotation impossible. Because this peptide bond is rigid, the backbone only bends at 2 single bonds flanking the alpha carbon, the phi and psi angles. These 2 microscopic hinges are the variables responsible for the complex 3-dimensional architecture of every protein in nature. Translating the 3D rotation of the phi and psi angles onto a 2D graph created the Ramachandran plot. Data is color-coded by viability. Solid green zones highlight allowed regions where atoms rotate freely. Red areas show disallowed regions where atoms collide, causing steric hindrance. Coordinates in the bottom left, known as quadrant 3, show negative values for both angles. This stable region is the home of the right-handed alpha helix, the most common secondary structure in proteins. Moving to the top left, or quadrant 2, we see the coordinates for extended backbone configurations. This specific spread of angles maximizes the distance between atoms, creating beta sheet structures. Every functional structural motif in biological organisms corresponds to a precise geometric coordinate on this map. While most amino acids fall into these zones, nature relies on specific exceptions to create flexible biological designs. Glycine is the simplest amino acid. Because its side chain is just a single hydrogen atom, it produces almost no steric hindrance. On a Ramachandran plot, its allowable green zones expand across almost all 4 quadrants. At the other extreme is proline. Its side chain loops back and bonds to its own backbone, forming a rigid 5-membered ring. This structure permanently locks the phi angle around -60 degrees, confining its allowable coordinates to a narrow vertical sliver. There are also intrinsically disordered proteins. Because they do not fold into a single stable shape, their coordinates fluctuate, scattering widely across the allowed regions like dynamic dust. By charting the boundaries of physics, the Ramachandran plot reveals how these specific molecules provide proteins with their functional diversity. Early structural biology relied on equipment like this vintage X-ray diffractometer to manually calculate atomic distances. This flowchart outlines how those early methods evolved. Today, high-resolution techniques like X-ray crystallography and cryo-EM generate complex 3D models, but they all still rely on the Ramachandran plot for structural validation. When scientists map a new protein, they plot its angles. If too many of those residues land in the disallowed red regions, the model contains overlapping atoms and is rejected as inaccurate. the Ramachandran plot remains a primary gatekeeper for modern protein engineering and drug design. To verify your understanding, consider this structural test. Which of the following statements about the Ramachandran plot is false? The false claim is that green areas indicate collision. As Ramachandran's Hart sphere calculations proved, the green zones represent regions free from steric hindrance. The red zones indicate the destructive collisions. Understanding these phi and psi angles lets us predict every protein's architecture, mapping the construction of life up to the scale of the entire planet. Simple geometric constraints at the microscopic level dictate the complexity of all biological organisms. Every protein in nature is the result of navigating this narrow set of physical boundaries.